Yeah, so as most of you probably saw, we're starting off seminar this term with short talks by faculty. This was a suggestion from the graduate students. If you're new and don't know who anybody is, I'm Matt Barber. I'm the one sending the emails about seminar and other stuff. Hi. So yeah, we're gonna jump right into it. The format, we've got up to 6 faculty talks, 5 minutes talks, 5 minutes for questions. I'll try and keep us on track. And I'm going last, so if there's no time left, I'll just be really fast. Okay, good. All right, so without further ado, we're starting off with Lauren Ponisio.
Hi, everyone. Here is our lovely Ponisio lab. So I thought today I would go over a broad, just like what we work on and then all the different projects going on in the lab so you can ask the people who are doing them what they're all about. So generally our lab has 2 main goals: understanding how species interactions contribute to species diversity and understanding how to manage and restore that diversity. We focus a lot on this idea of community assembly, or in the case of when everything got destroyed, reassembly. And we work with mutualists, particularly bees and their floral partners, as well as their fun microbiomes, whether parasitic or not. And we usually have a perspective on post-fire communities. So we work a lot in post-fire communities in the Cascades and around the state, as well as thinking about restoration. So our first project focuses in the Cascades and Northern Sierra wildfires. If any of you were around in 2020, those very convenient replicate fires for us. And Rose has been working on whether we can add floral enhancements into these landscapes that can benefit bee communities. Lauren Berger, our new master's student, will be looking at how plant communities respond to the compounded disturbance of stands being harvested right before a fire and then fires. And Rebecca has been working on the biogeography of the microbial communities and the response to high severity fire. Similar project will be working in Mount Pisgah. This is kind of a nascent project with the Dietz Lab and Lauren Hallett. And we've been trying to think about how we can design seed mixes to structure plant pollinator networks in specific ways and how plant pollinator networks respond to these controlled burns that Pisgah does every year. Jacob Swanson, another new student, will be leading that. We also work in beautiful, beautiful high elevation meadows in the Cascades. They're absolutely gorgeous. If you guys have been up to H.J. Andrews, and there we're looking at how interaction networks structure both long and short-term fitness of bees and their flowering plants. Nicole has been leading this work. And we've also been, measuring some things about parasites and stuff. Megan will be bringing a bunch of cool new information about movement as well. And so we're going to potentially be using camera traps to look at the movement across these meadows. This also caught on fire, and so it became some of our post-fire work. That was very convenient. Half of the meadow complex is burned, so we'll be understanding how fire contributes to all these things. We also have another gorgeous study system up in the Madrayanska Islands. This is a NSF grant that's coming to a close, so we have all of our beautiful NSF data. But basically, everyone in the lab is involved. Rebecca's working on the microbes, Nicole's working on the parasites. Alejandro's been studying how climate affects plant-pollinator interactions. Alejandro also is leading work that was recently funded by the NSF, and that money was not taken away, so that was a big win, even though the word climate change was used. And so we have these big transects across California. They were done in the 1960s, and we're resurveying them. And here's my favorite interaction network from Jasper Ridge Biological Reserve from the 1960s. There are all these beautiful bees. If any of you guys have been down to California grasslands, yeah, all those bees are dead, for sure. So we'll be measuring how dead they are, and that'll be really exciting. And through all these projects, we really need to be able to accurately measure bee communities. And so our new postdoc, Nevin, is going to be working on non-lethal methods for monitoring bee populations. We have these adorable little camera traps that point down, and we're using computer vision to identify the different species. Our lures, like our fake flowers, look way better now. These were like a first iteration that computer scientists made. Now they actually look like flowers. And as a quiz, our cute little lab logo has the most common bee in Oregon and the most rare bee in Oregon. Can anyone tell the difference? Which is which? Does anyone know which they are? Vozdesensky is the one that's flying in, and Occidentalis is the endangered bumblebee. So they look identical, and that's why they're really hard to tell apart. And so, yep, that's all the projects going on in the lab. And please ask the people in charge of them about them. And I will continue to do admin.
We can start with training questions. I think that's a good tradition.
You can tell the bees apart because Occidentalis has a little white bum. Just so you know. You see that bee, get really excited. And yeah, okay. Bosnian guy doesn't. Yeah, Emily. Why are all the bees in California dead? Ooh, well, particularly the serpentine really decreased. So serpentine's a really special community, and it's really restricted to very tectonic areas around the Bay Area, beautiful places to build homes. And so like after the 1960s, they really just urbanization took off like crazy. Climate change is probably playing a role. And then, you know, Jasmine's work, man, the invasive grasses are punishing. So these big highways led to a lot of nitrogen deposition, and that led to these invasive grasses establishing. So I think all the flowers are dead. And so like the flowers are less affected about the bees. Like they can probably do fine without them. But once the flowers are gone, the bees just disappear. Yeah. Most of those species have never been detected like in the serpentine for like the past, yeah. The last 50 years in California were a brutal time for urbanization. I mean, we haven't tested it yet, but I'm pretty sure. I was a cute little undergrad working at Jasper Ridge, and I did butterfly surveys out there. I never saw any cool bees. Yeah, Jordan.
So do you think that those
I think they're probably locally, I mean, you know, so where we've done most of our surveys so far have been in the places that are cheaper. So Jasper Ridge is really expensive to survey because it's in the Bay Area now. So until we had the grant, we haven't really been doing systematic surveys there. But for example, in Southern California, which is where this transect is, most of what we've caught are the European honeybee. And we've just not seen a lot of those bees that were probably already a little bit rare in the communities, maybe a little more specialized. All of these areas are intact, like they still exist, but like everything around them has changed. And so, you know, they've just completely lost all their floral resources and habitat. And yeah, the honeybee was almost completely absent from these surveys in the 1960s. Like it shows up every now and then. This is an invasive bee introduced to California in the late 1950s, probably. And it's just completely taken over these ecosystems. Whereas before it was like, like a couple individuals. Now it's like half of our surveys are the European honeybee. Yeah. It's an unfortunate common thing seen in like, particularly in southern, like more warm, they have more temperature tolerance. So they're able to persist. Yeah. Yeah, Biddy. One can dream. I did go out with the Oregon Bee Atlas as part of one of their field trips, and we got to survey serpentine, and I caught a bunch of really cool specialist bees. So you guys should all go down there in the spring, catch bees. Seems like a good field trip. Maybe we'll do it as an institute. Okay, thank you.
Yeah, I'm playing around with the order. Yeah. I think I've got Roshni. We're gonna do a quick swap from Keynote to PowerPoint. Do you have the little pointer thing? Here we go. Okay, so there's a laser forward back if you want to use it.
That's higher.
There's a tap on it. Is it on? May not be on. Just let's check. There's a little button here right on top. Now it's on.
And now.
Yeah, it can be a little bit higher, but it should be.
Okay.
All right. All right, next speaker is our newest faculty member in I Squared, Roshni Patel.
Thanks, guys. So, you know, as Matt said, I'm the newest one and I'm in data science. So I thought I would just give you a really general overview of the kinds of things that my group will work on and think about. And so the work that I do really sort of bridges evolution and population genetics with what I consider to be sort of like mainstream human genetics. The sort of central project in human genetics has been, mapping the genetic basis of complex traits and then trying to use that information to predict disease risk for individuals for things like asthma, heart disease, and so on. And, over the last 10 years in particular, our data sets have doubled and tripled in size to this extent that, the sort of the run-of-the-mill analysis in human genetics is now conducted in upwards of 100,000 individuals. And in this kind of data-rich environment, it has become really obvious that there's only so much progress that we can make towards those goals without thinking seriously about where genetic variation comes from. And by where it comes from, I mean how genetic variation is shaped by demography and natural selection, how it's structured within populations and across populations. And so the work that I do really tries to bring population genetic theory and methods into tackling these sort of bigger goals of human genetics. And one reason in particular, oh wait, let me just try this. Okay, that's fine. I can just use the same. Okay, so one reason in particular that this is super interesting to us is that kind of the status quo in human genetics for a long time has been all of the statistical methods that get used to analyze data, whether for discovering causal variants or predicting traits and disease risk. All of these statistical methods are kind of designed to be applied to relatively large and genetically homogeneous cohorts. And that's not super useful for us in trying to analyze diverse cohorts. It's not going to be super representative for understanding traits in global contexts. And so what population genetics gives us is a really principled way to sort of move beyond this paradigm and actually try to develop methods that are going to be more suitable for diverse cohorts. And I think you could argue that this is important for two reasons. One is through this sort of equity lens of this, moonshot goal of precision medicine. But I think it's also incredibly important from a basic science perspective because diverse cohorts tell you about how genetic variation arose in different evolutionary histories, about how traits manifest in different environments, different diets, different kinds of exposure to pollution. And so, yeah, some of the specific things that I think about in particular is how this sort of genetic architecture of complex traits gets shaped by selection and the role that the environment and gene by environment interactions play in this. But in general, most of the work that I do falls under this sort of umbrella of using population genetics to help develop smarter and better statistical models for human genetics. And then, in parallel, another sort of thing that I think about and work on is the social and ethical implications of human genetics. And this is in part motivated by the fact that this field has a really ugly history of, excluding data from underrepresented groups and also exploiting data from underrepresented groups. And we haven't fully escaped that legacy in the present either. In recent years, human genetics has been, co-opted and weaponized by extremist groups and race scientists and so on. And so, I think as this field moves towards working with more diverse data sets, it's super important to do that in a way that's mindful of the social and ethical implications. And so this work sort of looks a lot like meta-science or science of science research, and we collaborate a lot with bioethicists to answer questions about what are the social impacts of the research being done in human genetics and what are some resources and strategies we can develop to enable researchers to be doing more socially responsible research. So yeah, that's it in a nutshell. I do pop gen, complex traits, ethics, and always happy to talk about any of those things.
You collaborate strategies for ethical considerations or conversations?
Yeah, I mean, I think personally, I feel that sort of the biggest strategy one can take is actually to take this seriously as a part of education, and to every student that goes through a human genetics PhD program like needs to be taught all of this, like, and taken as seriously as like, you know, an introductory genetics class. But yeah, I think there's also some things that we have sort of in the pipeline, sort of related to like review at the level of like journals or like sort of what kinds of materials would you give a peer reviewer to help them understand like how the way that people use terms like ancestry or population or race are getting interpreted, like that. Yeah. Yeah.
Thinking about, you talk about a lot of classical human genetics, disease, history, populations, like mentioned migration. I mean, is there hope to learn something about like through migration history, like as people move to different environments, exposed to different environments, actually changing things like disease susceptibility? I guess we need like enough resolution of history of disease.
Yeah.
So recent migrations are like the power that people.
Yeah. So I guess it's kind of hard because for, I think those sorts of things, you kind of both want temporal genetic data and also maybe some kind of like temporal environmental resolution, which at the moment we kind of only really have in like great quantity for Europe. And I think we've like done a little bit of this, or like not me personally, but like people have done a little bit of this. And there's not like, you know, outside of the like handful of like large selective sweeps that we know about, or like the like, yeah, examples that we know, like lactase persistence and all that. For like complex traits, there aren't a lot. The selection has not been that strong. Like it's strong enough that you have to think about it when you're like, you know, trying to do these like association studies and all of that, but it's not so strong that it like, yeah, it's generating noticeable differences, the scale of those migrations, which is like only, I don't know, yeah, 100,000 years max. Yeah, for generations. Yeah. Cool. Yes. Oh, that's a very sweet question. I don't know. I guess I'm pretty excited for like all of the different hats that you get to wear at the same time. Like, you know, the research, the teaching, and the mentoring. It's a fun job to be able to get to do all three. Yeah. 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 All right. Thanks, guys.
I think I've got Biddy next. Assuming. No, I lied. Sorry. This is what happens when you wear all those hats.
All those hats. Well, I need this. Can you just project? Maybe.
Okay.
Yeah.
You've got the right shirt for it.
Good. Hi, everybody. You all hear me okay? My name is Lucas Silva. I'm a professor of environmental studies and biology here at URO. I've been here since 2016. And I direct the SPA lab, the Soil Plant Atmosphere Research Laboratory, which began at UC Davis when I was there and moved the lab to University of Oregon back in 2016. Here's a photo of our lab group in spring 25, missing a few people, some people graduated, other people coming in. I was debating for this, whether or not to talk about ongoing projects in the lab, or just to give you an overview of what we do and why do we do these things. So first, this idea of soil-plant atmosphere interactions as a core foundational concept in ecology and evolution is not an invention of my own. It's something that has been going on for a while. We use it in a way to address what I think is one of the most longstanding problems of pattern scale in ecology and evolution. And we do so by looking at mechanisms that regulate carbon cycling, that is fluxes of carbon, whether or not it stays in ecosystems, focusing on terrestrial ecosystems. And we do so across a variety of different ecosystems. We get funding from diverse sources of funding, and more recently we've been doing specific projects directed and funded by philanthropy dollars trying to work on climate change mitigation and adaptation. So there are many examples of the problem pattern and scale in ecology and evolution. Maybe the most relevant for this talk and for what we've been working on is embodied here by Simon Levin's 1992, the problem pattern and scale in ecology paper. I'll read here this quote from the abstract of the paper. He leads by saying, this is again before climate change was cool and before ecologists and biologists having funding to work on climate change and climate solution type thing. So the prediction of causes and consequences of climate change require the interfacing of phenomena that occur on different scales of space and time in ecological organization. The key to prediction and understanding, which is still an issue in ecology, lies in the elucidation of mechanisms that operate at different scales than those on which the patterns are observed. In other words, you measure some stuff in some scales, you try to scale it up and make that understanding of ecological processes and interactions predictive in a way that may be relevant for landscapes and the Earth system. And that is a challenge. I would argue that the old problem is still a persistent problem. It is one example. A paper came out in Ecology and Evolution, and I think this is 2018, the spatial temporal domains of modern ecology. And I adapted the data for this figure here to try and make it simpler. If you look at these two panels, let's begin with panel B, as you see here to understand here. Panel B is spatial resolution of the vast majority of data that we have gathered in recent papers and papers in ecology and evolution. And you see the distribution of the spatial resolution here. The distribution is the percent of the kind of scale resolution people are looking at. And as you can see here, the small plots in the field, if you ever done a grassland survey, a plant ecology type course, that's the most of the vast majority of the representation of ecological data that we have gathered over time. If you transfer this to temporal intervals, at what scales, temporally speaking, do we look at ecological processes and interactions and try to make sense of it? The vast majority is named here as unreplicated, meaning single sampling, single observation, and then move on. And for the replicated measurements, you see the month and weekly resolutions on the temporal side of things. That's why we have most of our ecological knowledge interactions coming from that sort of data. So with in this one example, we can point to major knowledge gaps, and my argument here is that one of the next challenges for us is trying to translate what we know from the small scale observations to more of a decadal to centennial and more of a landscape to regional level type of understanding and prediction, right? And how to do that? Easy to point the problem, hard to solve. We've been working on this thing that we call a predictive scaling framework, and we're not the only ones working on that. You can think of metabolic scaling theory as one component of that. The way we do it at the SPA lab, we try to link many scales. So we have different folks in the lab working at different scales here, all the way from in green molecular synthesis, biosynthesis, isotopic composition of certain signature molecules that we extract from organisms and gather signals from change in the environment and adaptation, things like changes in water use efficiency, carbon use efficiency, and so on. We look at how those processes forming those molecules, those molecular signatures, they explain changes in stomatic regulation, carbon-water exchange at the leaf level as well as the organism level, and how those might interact with things like root microbial associations and how those affect diffusion of nutrients and efficiency of use of carbon as it cycles through terrestrial ecosystems. From there, we look at how above-ground traits and how those mediate competition facilitation mechanisms that confer stability to certain types of communities under certain kinds of conditions, how the below-ground traits and trade-offs related to above-ground and below-ground traits then control resource acquisition, rise of scale connectivity, and so on. And at the scales of that management and policymakers care about, we try to make this leap through the whole ecosystem processes, thinking about soil formation, whole ecosystem carbon cycling and carbon sequestration, as well as whole landscape level trade-offs in carbon assimilation and water, for example. Huge, vast possibility for researching here, but what orients our research in this realm is really try to find causal relationships and things that can be predicted across these various scales of space and time. And if you're curious about how we go about doing that, testing theory to improve management, restoration, and conservation efforts. So this is the applied angle of our research portfolio. We have a vast network of collaborations. We start here and just showing the main regions where we have active research partnerships going on. Those are academic partnerships, as well as community partnerships, NGOs, and so on. Some of these sites and regions, or partnerships I established during my graduate training, all the way up to 2011. And then since then, shown in blue, the main regions of focus for us now are in northern Brazil and Pacific Northwest. There's two model systems, vast amounts of carbon, vast amounts of species diversity, and a lot of parallels between those two regions. And if you're curious how we actually do this stuff in the field, come check out some of our sites. We actually do experiments with backhoes. If you've never driven a backhoe, you can come to the Star Lab and do that. That's a photo from a soil science field trip we just did last week with undergraduates in environmental studies. Folks have an opportunity to actually do some, hands-on and experiential learning. We've also been around caves and reconstructing climate using speleothems and linking that to ecosystem change above the caves. That's a photo of central Brazil. We also do, of course, a lot of lab work and some molecular work in partnership with our partners, and try to make sense of it all using a combination of remote sensing, process-based, and data-driven models. That's kind of where our recent projects are trying to bring this all together. So that's it, I think, in a nutshell, what we do, and hopefully it was less than 5 minutes. And thank you all for your time, and let me know if you have questions. And yes, it says spa, so we don't have lab coats, we have lab robes. Katya is responsible for distributing the merch. Yeah. Thank you, Katya. All right. No questions? Yeah. Oh, yes. Sure. Thank you. Yeah.
Cell systems that you said.
Sure.
But beyond that, probably.
Sure.
Could be a lot of similarities.
Yeah, I could talk about the differences for a while. I'm trained as an ecosystem ecologist, right? So, in that kind of training, the emphasis is to look for the generalizable processes, right? So the data sets are not transferable or translatable, but the processes and mechanisms might be. So, the joke is that everybody passing through this biolab ends up being system agnostic in the sense they might have, preference for one ecosystem or another, but, the kinds of things we measure and the fundamental trade-offs, for example, carbon water fluxes, right? They can be translated and across not only scales, but also the systems. About the northwest of Brazil and the northwest of the States, one thing that is people don't realize is the northwest of the Amazon basin is maybe 9 times bigger than the northwest of North America in terms of what it encompasses. But the stories of ecosystem change and of people's ingenuity in both systems, the largest carbon sinks in their respective latitudes, the largest amount of biodiversity in their respective latitudes, the largest amount of water being recycled through ecosystems in their respective latitudes. And at the same time, similar problems right now with changes in climate, fire effects on those landscapes, and the social, cultural things that have yet to be discovered. And there's a lot there. I could talk about this for days, maybe over a beer would be easier. But anyway, you are all welcome to come visit the lab. If you haven't had a chance yet, we have a bunch of cool people there. We're happy to chat about this stuff. Thank you very much. Yes. Short distance, yes.
Okay, is this working? You guys can hear me?
Yeah, good.
Awesome. Okay, so I'm actually retired. I retired in 2019, but I can't quit doing science because I like it too much. I'm not teaching anymore though. So this is the last grant proposal I ever expect to write. And what we've been doing is looking at the distributions of fungi and quantifying ecosystem and climate drivers of their distributions. Fungi have, not only are there little penicillium and aspergillus, and yeast, which are pretty important, but also all the macrofungi that we know and love. In terms of people in the lab who've been involved in this, Jeff Dietz, Haley Burrell, Carolyn Delovich, and Heather Dawson have all been involved a lot. And then there's many co-PIs from outside. Okay. Remember, fungi perform a huge number of ecosystem services. Pathogens, for example, reduce plant biomass and they change plant community composition. Mycorrhizae increase plant biomass, okay, so they're kind of the opposite of pathogens, and they store carbon in the ground. Okay, so those are two hugely important functional groups in our forests and everywhere. Okay, so their distributions we know are going to depend on functional groups. Are they pathogens or mycorrhizae or something else? Climate, obviously. Habitat type. So oaks versus grasslands, for example, have completely different fungi. And where they're located. So soil is one sampling compartment. The air is a sampling compartment. It's not a substrate. They're just up there. They're moving around. And then, of course, the leaves and the roots and spore carps. Okay, so there's lots of different ways you can sample fungi, and what you're going to find depends on how you sample. Okay, we have a crazy experimental design. Talk about scale. So we had 8 long-term ecological study sites across the US, from the Pacific Northwest to the Southeast, Northeast, and a couple in the middle, and the desert Southwest. At each of these sites, we set up plots in grasslands, oak woodlands, and pine forests, Pinaceae. Not all of these habitats were available at all sites, but most were. And we also tried to do fire and no fire. Okay, we were getting really complicated. I'm going to ignore fire for the rest of this talk, but you can ask me about it. Okay, at each site we had these plots that had 9 subplots, and then we sampled each of these compartments from each subplot. Okay. I actually want to show you some data because that's fun. So this is all of the data, something like 30,000 species, okay? Remember that there's somewhere between 3 and 11 million fungi, and we only know about 150,000 of them by species, so we're swimming around in the dark a lot. Okay, so what we've done in this graph is the compartments, so where we sampled from here, and so most of the species were in soil, and then this section is replicated here. Okay, so soil and litter had the most fungi. And then in terms of where there was overlap, that's what these spots connected by lines are. So if you look at the all, these are fungi that were in all the compartments. And guess what? There were only 368 of them that crossed all the different compartments. So very much so, the species are compartment dependent on where they live. And this graph is made by Marcos Kaifa, who's a postdoc in the lab. Okay, now I'm going to get smaller and just talk about soil and litter because it's a lot of data. This is a multidimensional scaling graph. Each point is a sample. It's also a community. The closer in space the 2 points are, the more related they are, the more similar they are. And in this case, the colors represent the regions, okay? So an ellipse is surrounding the samples from a particular region. And you can see very clearly that region matters. And then within regions, the little cone-shaped things, which should be conifers, but for some reason we made them grasslands, are quite separated from the conifers. Sorry, from the oaks, yeah. So region matters and vegetation type matters a lot as to who's there. Okay, another way to look at this is to do pie graphs. These are pie graphs by functional groups, and I'm just showing ecoregion, not habitat type, but they also varied statistically by habitat type. And one of the things that you'll notice is that there's this giant pink wedge, which are the unknowns. Okay, so these are all fungi with no names that we know nothing about. So we can't put them into a functional group. And I found it really interesting that there were a lot of them, more of them in the Alpine in Colorado than in the Pacific Northwest. And also, of course, Florida being our southernmost, most closest to tropical, had lots of unknowns as well. Okay. And finally, just a quick compartment and habitat type explain the most variation. This is a heat map that Jeff and Haley made. So on this axis, we have various richness and diversity, like exomicrorhizae and litter. And the darker the color, the larger the effect size. These are all significant. Okay, every spot up here is significant, but the color tells you the effect size. Okay, for this one, ectomycorrhizal richness is strongly affected by whether it's in the soil or in the litter. Basically, this is soil. Okay, they're only in the soil. They're really not in the litter. Whereas plant pathogens down here are negatively affected by soil because they're only in the litter. This makes sense, right? A lot of plant pathogens are in the leaves. And so when they fall down, they're in the litter. Okay, so one of the things that surprised me about this heat map was that I thought this was going to be much brighter than it is, because traditionally people have thought, well, pH is the main driver. No, with fungi, it's the plants that are the main driver, right? It's compartment, so litter versus soil, for example. This is just litter versus soil. And the plant communities, whether they're in oaks, grasslands, or pines. So I'm going to stop there and let you guys ask questions, but we're actively working on this. We've just gotten all the data together and it's just exciting and fun. Yeah.
Just curious about, I think it was like, but they just like so stick out a lot in the NDS. Yeah,
Yeah. So what are you looking at these red ones? Why are they?
No, I was looking at those like blue ones in the top, the oats.
These guys up here. Yeah. I have to look at those two samples specifically in order to answer the question, but fungi do move around. You know, one thing we measured spore dispersal, and it was most of the things that we saw we knew grew within about 40 kilometers of the sites, but there were some way out outliers too. So yeah, Lauren.
Can I ask you the kind of opposite of the Roshni question? Like, what after, you know, theoretically retired in 2019, building that eventually happened, but what did you enjoy most about all the years of ecology and evolution?
Oh, the students. Yeah, I mean, I think probably the journal clubs, frankly. Talking, you know, just getting to know people and talking science. I think I enjoyed that the most. What I found hardest is what you're just beginning to learn, which is the multitasking, the trying to be good at all things all at once, all the time. And that was the benefit of retiring, is that I could focus. Andy?
Yeah, so pH isn't structuring things.
No.
Plant communities are.
Yes. What's structuring the plant communities? Partly pH, absolutely.
Well, is it secondary? Structuring the fungal communities then?
Yeah, I think it is. And we see that really strongly. For example, conifers are fairly nasty. They're full of lots of secondary compounds. And they have thin leaves that are hard to land on, and they have fewer fungi. Yeah, and the ones that have the most are the oaks with their nice fat leaves. And those communities also include grasses, so they've got both. Yeah, great question. So Lucas and I should talk about these secondary. Okay, good, thanks.
Next, we have Andy Kern.
I never see those things on. Thanks.
How that works.
I do.
This should work if you want to use the laser or whatever.
I will. They want me to turn around because shout out to Clara, she made me this awesome shirt. I think of this as my Eric Adams shirt. If you all know who Eric Adams is, the mayor of New York City, who famously wears a New York City polo office of the mayor, and it says mayor on the back. It's freaking awesome. Quiet. In my new hat, my new role, I want to point out a couple things. First, this is an awesome turnout for a kickoff seminar. Second, I think there's a sign-up sheet going around. Awesome. Please sign it if you haven't. We want to start getting a metric of like attendance on these things moving forward. Cool. So I'm going to give a little flash talk, and I'm not going to do, so Biddy kind of did this. So I'm going to talk about one story. I could talk about all the things that the lab does, but that would take me more than 5 minutes. So instead, I decided I was going to focus on one story of a project that I'm working on right now. Generally, one of the threads that goes on in our lab is we are machine learners. So we are applying modern machine learning and deep learning techniques to population and evolutionary genetics. I said population genetics here. And so what that looks like generally is something like this. We take as input genotypes, so like DNA sequences. We pass those through deep neural networks, and we try to predict things. And one of the problems with doing this, essentially, is that machine learning and deep learning over the last 15 years has really been driven by large tech companies like Facebook and Google and whatnot. And like the primary, before the AI revolution, the primary use of this, if you can believe it, was labeling pictures on the internet. So like if there's, you upload pictures to my Flickr, or maybe you know about Flickr, but to whatever your I'm dating myself, to whatever your favorite website is, and Google will troll through those and try to say, this is a picture of a dog, or this, where's my pointer? This is a picture of a dog, or this is a picture of a cat. And because they're interested in this labeling, what we call in technically point estimation, there are really less developed methods for saying how certain we are that this is a picture, or this is a picture of a dog. Like, are we 70% certain this is a picture of a dog, or 60% certain this is a picture of a cat? And this is problematic for someone who studies evolution. It's problematic because evolution as a process is this hugely noisy process. So when I have DNA sequences and I'm trying to use machine learning to make some kind of inference about what the heck is going on, I want to know how certain I am of that inference. And this is way less developed in the field. Oh, I'm too far away. Come on, Tucker. So canonically, the way that folks in statistics deal with uncertainty, or like the most popular method, is essentially goes like this. If we can write down some likelihood function for our data, like what's the probability of my data given some parameterization, call it theta. I can write down the log of this thing, call this the log likelihood function. There's this mathematical object known as Fisher's information, or Fisher's information matrix, whereby I can say, given the likelihood of my data, I can form some ellipse around my point estimate, given this mathematical construct. and I can say, oh, my point estimate is consistent at some level, say 95% certainty, with these other estimates. Okay, so all of this is contingent on being able to write down the likelihood of the data in this model. God, I keep walking too far away from this. But increasingly, what we've been doing in the lab, we and others as a field of population genetics, is we've been focusing not on writing down mathematical models, but instead using some simulator that basically is a stand-in for a model, whereby I can parameterize my simulation with some value here, plug it into my simulator, and simulate some data. And this simulator implicitly defines a likelihood for me. Like I can do many, many simulations around some parameter and get many, many values of my simulated data out. And so we've been working in this worldview for our deep learning and machine learning. And the problem is that we don't have a way to write down uncertainty with our simulators. So you basically have two choices right now. One choice is to simulate many, many times about your estimate. We call this a parametric bootstrap. So I might have some estimate. I do many, many simulations, conditional on, I have some data. I plug it into my, say, neural network. I get some estimate out. And I then do many, many simulations. And I can build up some uncertainty this way. This is very, very expensive, often computationally. Another way that I could do this is I could do some non-parametric bootstrapping. So I could, say, resample from my data many, many times, plug it through my deep neural network, get many, many estimates out, and, fill out some ellipse that way. But the problem is that while this has some guarantee that it should work, but it's expensive, this has no guarantees about whether we say my uncertainty is well calibrated, sort of ad hoc. So we in the lab, I thought I changed this starting. We in the lab have been working on a new approach that's come out of the deep learning world and the machine learning world, what's known as neural posterior estimation. In the interest of time, I'm not going to go too far into the details, but all of this depends on a kind of neural network called a normalizing flow that basically allows us to fit some target distribution, like the uncertainty around some point estimate, from some easier to sample distribution. And the way this works essentially is that if I have some data, I plug that into some kind of embedding network, some neural network that might take some kind of shape. I get some point estimate out from this. I plug this then into a normalizing flow of this different kind of neural network, along with some kind of noise. And in so doing, I'm able to get an approximate posterior estimate of my parameters conditional on the data that I put in. The beautiful thing about this setup is that I can use neural networks here that can automatically extract whatever are relevant summaries of my data, plug it through this whole giant mousetrap, and at the end get out some nice uncertainty. Whoops. I should say this is work that I've been doing in the lab with Nate Pope and a former postdoc, Ji-Sun Min. Here's an example of what this looks like. So we previously developed a neural network method for estimating recombination rates from genetic data. That neural network method worked quite well at that point estimation thing, but we didn't have good uncertainty estimates about our recombination rate estimate. So we relied on parametric bootstrapping, which I should say is kind of like the gold standard, but I already mentioned it's really, really expensive. So we can do this, say, for a whole bunch of recombination rates. We can get parametric bootstraps out using our old method. They kind of look like this, about the true mean. We can also apply this posterior estimation routine, this neural posterior estimation routine, and we get essentially the exact same posterior out. This is much faster than this, computationally less expensive, and we can show that the coverage is nearly identical. So we're essentially getting all of the goodness of parametric bootstrapping out through this much more efficient routine. We can apply this broadly to all kinds of problems in population genetics. This is a figure from the paper we're working on right now. This is applying this idea to trying to estimate population size. So this is population size on the y-axis, over time. These are three different histories, one where there's an abrupt change, one where there's a gradual decline going back in the past, and one where there's a gradual increase. And I'm showing three different embedding networks. So these are three different neural networks that I use to extract information from the genotypes that I pass in. One is called the convolutional neural network, one is called a recurrent neural network, and this is a bespoke kind of network. And we can see that our posterior estimates generally include our true parameters throughout this history, and we get these nice uncertainty estimates associated with our point estimates. Okay, so I'm going to finish there. Hopefully I kept it to 5 minutes. These are the folks that I'm thanking here. Did I not keep it to 5 minutes? Okay, I get one question. Neural networks. Yes, Kendall.
Uncertainty and like treating to like kind of pick it up and like you don't know what it is like there's like any is there any relatedness to like what you're doing and like when we do those like I am not a robot personalities?
Well, there is. Okay, here's there's no relationship between that and uncertainty as far as I know. But do you know what that's doing? That's the companies getting humans to label data.
Yeah.
Like literally, that's companies, that reCAPTCHA company is selling that to like Google or something else like that. So you are the mechanical Turk, as it were, labeling data for tech companies every time you do that. I fail those all the time, but which makes me question my humanity. Okay, I'll be talking to you all later anyway at the retreat. So can I make two more announcements? So one, I already said this in email that you guys already got. Coffee and cookies, that's going to happen before every seminar. After seminar, moving forward, starting next week, Matt is giving seminar. Moving forward, after seminar at 1 p.m., there will be a trainee lunch with the speaker. That trainees include graduate students, postdocs, and everyone that's not faculty. You all will be welcome in 318 for pizza with the speaker, to have time with the speaker for an hour without your annoying PI around dominating all the time. So those are my two announcements, and I'll see you all at the retreat later.
Need yours. I'm going to do one.
Oh yeah, Matt's got an announcement.
No, I got to give a talk.
Oh, Matt's got to, oh, dude, I'm sorry.
You're good. You're good. So I always feel bad asking faculty to give talks if I'm not willing to do one myself. So like, that's part of it. I also put myself last, knowing that somebody's going to go a little bit long enough. I will be really fast though. And also Andy saved me giving some end of seminar announcements. Yeah, so thanks everybody for coming. This is a photo of my lab from the summer. Amazingly, like 4 of these people have now moved on to jobs in grad school and other stuff. But in my lab, the big questions we're asking right now, I think basically fall under these 2 things. One, we're curious about how animals have evolved in order to defend themselves against pathogenic microbes. In our lab, we think a lot about pathogenic bacteria. And secondarily, how do these bacteria themselves evolve and adapt to the challenges that they encounter in an animal host environment? And so this is our favorite bacteria that we mostly are working with in the lab, called Staphylococcus aureus. About a third of the people in this room have this bug in their nose or on some part of their skin. In addition to looking like cute and like a little cluster of grapes, it's also a major cause of human infections and antibiotic-resistant infections, which is part of why we are studying it. So I'm just going to tell you real quick about 3 projects that 3 grad students are working on in the lab, really just the questions that they're addressing. So Killian Campbell, who's defending his thesis next month, yay, has been working on understanding how staph adapts to toxic molecules that they encounter in the host environment. So actually, it turns out staphylococci are sensitive to these molecules called polyamines that virtually all other organisms make. And for reasons we don't understand, polyamines kill staph very well. And so he's been using experimental evolution to understand how these bacteria can adapt to these compounds that they encounter in a host. Kendall Tinney, also a current Grebes president. I think you need a shirt like Andy's. And president sounds better than director. So Kendall has been working on projects relating to phage, so understanding how phage and bacteria interact, but more specifically, how the environment that these microbes are in shapes their coevolution and sort of ecological interactions. So specifically, she's been working in a media that mimics the environment in which they're found, which is actually the nose. So using this synthetic snot, synthetic nasal media. And actually, she's found some really cool stuff about bacteria that are normally susceptible to phage in normal laboratory conditions. These viruses now are resistant when you put them in a media that mimics the host environment. And so she's trying to figure out a little bit about why that is, among other things. And then finally, Titosh Still is sort of working on the host side, that first question that I posed earlier, and specifically, how do animal antimicrobial proteins first emerge and later evolve? So she's been looking at actually how antimicrobial functions can evolve within other pre-existing proteins, and both how those functions first arise, and then actually specifically within primates, how natural selection has actually shaped the function of some of these proteins. So that's my really quick summary. If you have any questions, you ask me now. I'll have a beer at the retreat later. You can ask me then too. But yeah, we have a minute for questions, and I'll go. Thank you. Or if you go really long with questions, then I don't have to go now. Yeah, Clara. Do I know if I'm I have not swabbed myself, and I can tell you that everybody has some staph that aren't aureus on their skin, but not everybody has that species in their nose. But I don't know myself. Yeah, I don't want to find out that I have MRSA. That's what it is. You can ask me about that later. All right, thank you.